We are continuing with the session, which is in fact summarizing more of the clinical and developmental studies to get to more clinical understanding and treatments development. So the second speaker is Allyson Berent. Good to see you. Allyson is a veterinary internal medicine specialist at the Animal Medical Center in New York. Allyson's daughter was diagnosed with Angelman syndrome in 2014. And since then, she has been a force of nature behind developing so much to try to understand better Angelman syndrome and to develop treatments. She has been working nonstop. She's in many, many projects. I'm going to just mention a few. She's in the board of directors of FAST US. She's co-directing ABOM, which is another association that develops biomarkers. She's collaborating with a consortium of scientists working on translational research in the US and throughout the world. She's a co-founder of Gentex, which was acquired by Ultragenyx, and this is focusing on the antisense oligodeoxynucleotide therapy, as we have heard from Lynn. She's Chief Development Officer of the drug accelerator AS2 Bio. Maybe we hear about that as well. And so I'm just going to go to the title. She's going to give us a fast roadmap to a cure. Allyson, thank you.
Thank you, Christina. And Lynn, that was hard to follow. I'm going to do my best here, but that was an excellent talk, and I think very well received by the family. So thank you for walking us all through that very slowly and very elegantly. So I am going to talk about accelerating drug development for Angelman syndrome. And as mentioned, I have a couple roles. Firstly, I serve as the Chief Science Officer for FAST US. I am also the co-director of the Biomarker and Outcome Measure Consortium, which we'll talk a little bit about, and the International Angelman Syndrome Research Council. I do help with drug development as the Chief Development Officer at the accelerator AS2 Bio, which I'll talk a little bit about as well. I am a veterinarian by training. That's where I started, mostly in medical device development, running clinical trials in animals with the ultimate translation of medical devices for kids. And the most important job that I have is that I'm the mother to that beautiful little girl on the right. Her name is Quincy, and she lives with Angelman syndrome. Here are my disclosures. So I'm not going to talk about the genetics because Lynn already did a really excellent job. I'm just going to highlight a few things in which I'm going to walk you through a little bit more slowly as a parent, because sometimes really understanding this can be challenging. And starting from the basics, which Lynn did a great job of, repetition is always really good to truly understand it. And as Benedetta said, knowledge is power. The more you understand, the better you're going to do for your child. And that's really important because the decisions are yours, because our kids can't make those decisions. And so I want you to really understand things today, which means don't be shy, raise your hand and ask questions. That's why we're here, to educate you as parents, to be able to understand science, which is really complicated. And so we're here to break it down to the basics of what you can truly understand. So as humans, we all have 46 chromosomes, one from mom and one from dad. Mom is called maternal and dad is called paternal. And on one of those chromosomes is a gene of over 20,000 genes that is called UBE3A, as Lynn really nicely talked about. And if there's a defect in that gene inherited from mom, that results in the syndrome called Angelman syndrome. And that affects approximately 1 in 15,000 or 500,000 kids and adults potentially throughout the world. We don't know the population because they're not all diagnosed. So many are left undiscovered. So it is our job as a patient advocacy organization worldwide to find them, to ensure that they have what you have today, which is community. Because community is what allows us to survive as parents living with the challenges of Angelman syndrome. It is usually random, it is rarely inherited, but it can be, which is why genetic counseling is so important for us parents who might want to have multiple children. And I have 3 little girls. 2 of my children are typical, and my middle daughter lives with Angelman. Every chromosome codes, as Lynn showed you, for many, many genes, tens of thousands of genes. And generally, the genes code for proteins. And UBE3A is a protein. And it is very, very important in bodily function, as we all know. Because if you're missing it, you understand the symptoms of Angelman syndrome. So I'm not going to spend any time on this. This is maternal and paternal, mom and dad, chromosome 15. And here's the schematic that Lynn already showed you. Remember, the maternal copy is red, but only in neurons of the brain and peripheral neurons. The paternal copy is silenced by this RNA called the antisense transcript. That silencing results in you and I only having the mother's copy read. But our children don't have the mother's copy read. It's either deleted, mutated, or silenced, resulting in the symptoms of Angelman syndrome. And again, all of those genotypes I'm not going to talk about because Lynn already did a great job of that, but it can happen in many different ways, all resulting in unbelievably similar symptoms, regardless of how or why you have UBE3A deficiency on the maternal or mother's copy of the gene. So what does UBE3A do? Well, it's a protein, it's an enzyme, and the enzyme basically degrades or upregulates other proteins, particularly in the brain. And that control and that balance of different proteins ultimately results in allowing neurons, the brain cells, to talk to each other. It tags proteins to say you need to stick around or you need to go away. And this is required, this incredibly intricate and elegant balance for neurons to communicate well to each other. And if they don't, and there's this deficiency of UBE3A function, which results in accumulation of other downstream proteins, or the lack of other downstream proteins, you have chaos at the level of the junction of 2 neurons. That junction is called the synapse, and you can see that junction right here. And that results in a synaptopathy, or what we also talk about as tonic inhibition, which is something you may have heard. Tonic inhibition is basically where the communication between 2 neurons is overactive. So what happens? We have too much excitation and not enough inhibition. What does that mean? Well, if you think about the symptoms of Angelman syndrome, if I want to walk, I need to activate one leg and inhibit the other leg. If I want to talk, I need to activate my tongue, my voice box, my cheeks, the muscles of my head and my lips, and inhibit other muscles so that I can make sounds and I can communicate. If you don't have that intricate communication between neurons which drives muscle function, you can't actually perform. It doesn't mean you can't think, it doesn't mean you don't know, and it doesn't mean you don't understand. It means you can't express, it means you can't walk as well, balance as well, swim as well. That pretty much describes Quincy, right? She understands a heck of a lot, but she can't express herself, she can't walk well, and she has challenges with communication of her neurons and her muscles. That is the symptoms in the simplest of terms that we live with every day. So I'm just going to expand a little bit upon what Lynn talked about, the the deletion genotype. And the reason I'm going to do that, just a little bit, is because I want you to understand the drug development pipeline, and I want you to understand why we're developing certain drugs the way we are. So the deletion, we're missing a chunk of DNA on the maternal copy, and this affects about 70% of our population. If we break this down into a schematic, that deleted side on the mother's copy is expanded here. UBE3A is highlighted in red. This is the region that Lynn talked about that could be deleted. Type 1 or class 1 deletions, you could see at the bottom, is from breakpoint 1. See the NEPA-A1, that gene, all the way to breakpoint 3, HERC-C2. That whole region is deleted in type 1, where from breakpoint 2 to breakpoint 3 is deleted in class 2, to your question you raised earlier. Why does that matter? Well, that means that these groups of genes that make important proteins are also deleted. As Lynn said, they're not imprinted, meaning that they are expressed both from the maternal mom and dad. So we have 2 copies expressed in all of us of those genes. The only one imprinted is the UBE3A gene that only must be expressed from mom. Therefore, if we look at, of those 4 genes on the left and of these 6 genes on the right, what's important? Well, let's think about and let's dig into the genetics of each of those genes, and do we know about the function they have in humans, right? Are there humans with mutations in 1 copy of those genes and not both copies of those genes? A lot of the diseases associated with those genes that have UBE3A expression may still have some symptoms that we should expect will be left over after we replace UBE3A. But what's important to remember is that the symptoms in those individuals are far, far less severe than Angelman syndrome, which means if we replace UBE3A, we have every reason to believe we can make a tremendous difference in the lives of not only mutation individuals, but also every class 1 or class 2 deletion individual. But we're not going to stop there, right? We're here putting it all in. If we're going to put it all in, and we're going to really do our best to transform the lives of every single individual born and not yet born with Angelman syndrome, we have to think about how are we going to get it all back, if it's necessary, at all ages. Not yet born and born, and living every day with this disorder. So if we look at that list of genes on the left, which is highlighted above, what you can see of those genes is that some of them don't seem to have much impact if only 1 copy is missing. Some of them only have impact if both copies are missing, or if one of them has an overexpression. So when we really highlight what we think is most important genetically and clinically, really those GABA genes stand out quite a lot. But some of the other genes could have impact. There are reports where individuals have mutations in them and have some maybe developmental disability, maybe have some speech challenges, maybe have some seizures or a history of a seizure. Regardless, we want to understand it, which is why we're looking to also express those genes too, and I'll talk a little bit about that. But UBE3A is by far the overwhelming culprit in Angelman syndrome, and this we know based on what Lynn said. We know mutations, we know deletions, we know UPD and ICD have all of these common symptoms. I don't need to tell you guys the symptoms of Angelman because you live with it every day, as do I. But what's most profound is the universal lack of speech, the potential for debilitating seizures, and the lack of sleep. Our kids cannot live an independent life. That is the most profound. But let's not forget the impact that that has on you, on me, on family members, right? It's not just the symptoms to our kids, but it's the symptoms to the entire family village. Complete fatigue, inability to maintain work, stress, social isolation, giving up your life in order to help support your child because they are 100% fully dependent on you. This doesn't matter what genotype you have. UPD, ICD, mutation, or deletion, they are dependent on us adults for the rest of their lives. That is the most debilitating part of the disorder, and I think we can all agree to that. We cannot forget the impact it has on so many of us. This paper came out, and this is the first paper that I read after my daughter's diagnosis. And I'm going to be honest, this paper completely saved my life. The reason it saved my life is because what I realized is that there's a very small portion of kids with Angelman that have mosaicism that Lynn mentioned. Mosaicism means not every cell has a deficiency in maternal UBE3A. Some cells are expressing maternal UBE3A, meaning that the defect didn't happen at fertilization, at the egg stage. It happened after fertilization, after some cells were already normal or typical. Therefore, they have some UBE3A expression on the maternal side, but most cells have no maternal expression of UBE3A. So what this showed was a clinical correlation to the known peripheral, meaning blood cell expression. What percentage of blood cells are expressing maternal UBE3A? What percentage of blood cells are not expressing UBE3A? In hopes that that translates to brain, what neurons are expressing maternal UBE3A, what neurons are not. And we don't know that that's a direct correlation, but what this will tell you is that the more UBE3A you see, so on the right you could see that that's 0, 1 to 5% of maternal expression. On the left you could see up to 40%, and then it jumps to 90% maternal UBE3A expression. For kiddos that have 1 to 5% of normal functioning cells, they have some speech. They may not have seizures, and they can walk very, very well. 10 to 20% essentially have no seizures, can speak in phrases, maybe sentences, can text message, I've met them. And over 40% are often undiagnosed because they might be actually neurotypical, because we probably don't need more than 40%. What does this tell you? Well, it tells me 1 to 5% is a different child than I live with every single day, and I would do anything for 1 to 5% to change her life. So the enemy of good is better. We don't need 100% in order to improve the lives of those living today with Angelman syndrome. A little bit can go a really long way in making a difference in their lives. We are not here to say cure or nothing. We are here to say transformative treatments can make a difference in our kids' lives. And that's what we have to aim for. And over time, like everything in the world, it will get better and better. There will be a second generation, there will be a third generation, there will be a 10th generation, but we have to start with the first generation. And if we just wait for the best, the enemy of good is better, we're gonna get nowhere. This is like everything in life. We started with aspirin, and now we have lots of safe non-steroidal medications to help with inflammation and arthritis. Why are we waiting for the best? So this is why we have to make progress, incremental progress. And that's what we're trying to do here. So when we think about the roadmap to do that, which is our mission and our goal, is to make a difference, we can replace the maternal gene. We can replace the maternal protein. That's with a gene replacement therapy or with enzyme replacement therapy, just to keep it at a very high level. We can unsilence the silent copy of the gene, like Lynn talked about. We could do that in many ways. We could do that with ASOs. We could do that with zinc fingers. We could do that with CRISPR gene editing or with miRNA. So there's lots of different modalities to get to the same result, and they all have different opportunities for our kids. With gene replacement therapy, we can do that through an AAV virus. We could do that through a lentivirus, which we're going to talk about today. Or we can just put the protein back into the brain and let UBE3A function. All of those we're showing could have benefit in at least a mouse model of Angelman syndrome. What about downstream targets? What about the synapse? There's so many ways we can help the synapse function better and work better, and we'll talk about that as well. But we cannot forget, while we do all of this incredible, amazing science, we have to be prepared for clinical trials. We have to be prepared for drug approvals. We have to prepare our families to be ready to take this on, because this is a huge commitment, and it is absolutely required participation and collaboration with families and caregivers. So we need to be prepared for that. So let's start with how do we replace mom's gene. And I'm not going to talk about the science because we have other talks that are going to talk about that. I'm just going to give you a little preview that we can look at AAV gene replacement therapy by putting a virus that's a healthy copy of the virus into the brain through the spinal fluid, or even now potentially crossing the blood-brain barrier from the blood, and drop off a copy of that gene in neurons. Let it secrete UBE3A. We can actually do a bone marrow transplant of our kids' own cells where we modify them to put the healthy copy of UBE3A into those cells, where those cells actually disperse into the blood, cross the blood-brain barrier, get into the brain, and can feed UBE3A to the neighboring neurons. We're going to talk about that today. We can replace it by depositing the enzyme, the protein itself, into the brain. And this part I want to highlight a little bit here is we can upregulate the genes outside of UBE3A. We talked about the GABA genes, all of the other genes that are deleted. We have a program where we are upregulating those genes on the side in which they're not deleted, the paternal allele, because they're not missing there. And we can overexpress them to try to meet what's missing on the maternal allele. CRISPR activation. So we are addressing also the deletions from a genetic perspective as well. These are all the ways in which we can make huge benefit. So these are going to be talked about today. This is just data in mice to show that you can actually do this. It's not science fiction. And Jennifer's going to talk about AAV and lenti, gene replacement therapy. I just want to show you a video, because you don't need to see the graphs and understand UBE3A expression, really. I mean, we need to understand that as the scientific community. What you need to see is this. This is an Angelman mouse on a treadmill. Adult Angelman mouse on a treadmill. After that adult got a bone marrow transplant with UBE3A in it that crossed the blood-brain barrier and got to the brain, that's the Angelman mouse on a treadmill. This mouse got treated at 8 weeks of age. That would be the equivalent of an adult. It's a big difference that you can see in one mouse, pretty obvious. This is another video. This is a normal mouse, an Angelman mouse, and a genetically treated mouse with hematopoietic stem cell. It doesn't take a genius to see the difference. You could see that the Angelman mouse is pretty slow. Poor little fella falls off. And you could see all the other mouse made it across the balance beam. In a single mouse, you can see a dramatic difference. It's profound. This is where all the money of research goes in order to try to prove proof of concept. Can we do this and make a difference in mice? Because we have to prove that before we can make a difference in humans. Pillar 2, unsilencing. All of the different ways that you can unsilence. I'm not going to steal any thunder here. Lynn already talked about this, and we're going to hear a lot more about this tomorrow. The antisense oligonucleotides can rescue phenotype. Zinc fingers can rescue. You could see the brain at the top is a typical wild-type mouse. In the middle is an Angelman mouse, less brown. At the bottom, you've activated the silent copy of the gene expressing UBE3A. More brown equals better. Here's the mouse behavior, showing you can improve their behavior. So these are complex experiments that are being done, and Dr. Zilka is going to talk to us later about CRISPR technology. Dr. Yangwei Zhang is going to talk also about CRISPR technology. I'm going to let him go into the details of this, but this just shows the potential of his incredible work of being able to permanently CRISPR edit the paternal allele in order to unsilence UBE3A and allow it to be expressed in the mouse. This is a CRISPR from Yangwei Zhang's lab. You could see the wild type on the left, Angelman in the middle, and an adult CRISPR gene-edited mouse on the right. It's pretty obvious. It has a huge impact on a mouse. A mouse is not a human, and we must remember that. And then what about downstream targets, the synapse? How do we get at that? Well, we're going to hear from Christina Alberini about IGF-2 ligands. We have BDNF analogs, which can address the signaling between the two neurons. We can rescue phenotype through correcting oligodendroglial cells, which impacts myelination. And then there's a trial right now by Roche using alogabat, which is upregulating the function of the GABA receptor, which could be beneficial in deletions. So I just want to highlight really quickly this heat map. This is a heat map of mice. And I also want to highlight, as parents of children living with Angelman, your kids are not mice. Your kids are human. But the only way we can assess the potential for benefit, which is no promise of benefit, is to start with mice that have some behaviors that look like what our children experience. And they are not the same. Their brain is so much less complex than our kids' brains. So for anyone to believe what a mouse tells us is what a human is going to tell us is really mistaken. A human is going to tell us what a human tells us, but a mouse might help us predict. And that's what's really important about mice. So there was a paper that came out a few years ago looking at reinstating at different ages UBE3A and the impact that that could have over time. So if you look at the top, you can see before birth, prenatal, at birth, juvenile, adolescent, and adult. And you can see it gets more red as they get older, which led people to believe the earlier the better, which makes sense. As the brain develops, the earlier you give back the gene, the more rescue you're going to have. Very similar to learning a new language. If I started learning Italian today, it would probably take me about 3 years, and I would always have an accent. But you might be able to understand me eventually. If I learned it when I was 2, I likely could be fluent in Italian within 3 months and never have an accent. Why do we believe it's any different for other developmental things for our children to learn? They may never do it perfectly. They may always have an accent. But I'll take an Italian accent any day over being silent, over having no words. So let's not get stuck on perfection, right? Let's put it in, and we got to see what they can put out because we just don't know. Look at this. This is all of the programs from different labs using different modalities of therapeutic potential. ASOs, CRISPR, zinc finger, AAV gene therapy, and hematopoietic stem cell gene therapy. This is right out of the literature. What do you see with this heat map? It looks really different than the first heat map. This is not a reinstatement model. This is a gene transfer, gene-targeted model in the AS mouse, but the data's all over the place. You have a lot of green, you have some red, different labs look at it in different ways. Adults with full rescue with hematopoietic stem cell gene therapy, with CRISPR gene editing, non-viral delivered, and then you have some that have a lot of red. The ASOs have the most red, unfortunately. CRISPR seems to have a lot of green, Lenti has the most green, and AAV is absolutely all over the place. More green than red, but it's all over the place. What does this tell me? They're mice. That's what it tells me. It tells me that we have a lot of reason to believe we can rescue behaviors, and we don't know at what age, to answer the question about age. So if we look at pillar 1 and pillar 2, just to blow it up, that's optimistic to me as a parent. I also feel it means we can't put all of our eggs in the mouse basket, right? We have to move to humans because that's going to be the definitive experiment. And we have to use mice to give us prospect for benefit. They're not going to define the benefit. Who's going to define it? Fortunately or unfortunately, humans. That's the species that will define it. And they're doing that right now, thank goodness, in all of these clinical trials. So lastly, I'm going to end with pillar 4. With pillar 4, it's how do we get all of this science to humans? That's complicated. We need tools. Okay, we have more animal models than I care to admit. Honestly, as far as I'm concerned, we have to be done with the models. We have too many models. We have so many. We've spent millions and millions of dollars on models. We have 11 mice, 2 rats, 2 pigs. I think we're good on the models. We have cell lines. We have lots of cell lines. Those are truly and deeply important because we have to test this in human lines, right? So we have repositories. Not only in Italy has a biorepository we're going to hear about today, also in the US we have a biorepository, and I hope that we can collaborate. The 2 biorepositories made commercially available to everybody. Anybody can have access to these repositories through eBisc. And on our website, we have all of the bioresources that are available to all researchers around the world, that anything FAST ever funds is totally and fully available to all. Nothing is held proprietary. So everything has to be shared or we will not fund it. Therefore, you have access to anything that you need. We have a literature repository. You, parent, want to go in and research anything, it's there for you. You can research that. It's a really fun tool on our website for you to find papers and see anything you need to see based on how you want to search for it. It's really, really fun. We had to do a natural history study, and thank goodness for Lynn and Wenhan Tan and Liz, who are both here, who did this over 18 years. And Lynn showed this, but this is the output of that. Our kids going back every year to the hospital for these studies that we didn't know the value of have so much value. Because what it tells us is how do they behave over time, and will a drug surpass natural history? Will we surpass that to a point in which we don't have to have so many controlled studies where kids are not getting treatment in order to prove if a drug works? We will have to start with those controlled studies, but we want to borrow from natural history over time. The Biomarker and Outcome Consortium has been an incredible experience working with other advocacy groups, pharma, clinicians, as well as scientists, to try to develop what matters, what's meaningful, and what's measurable. When you say, why are we doing all of these assessments and these trials? It takes me 2 days for me to go to a hospital to have all of this stuff done. Because we determined, as we narrowed down all of the ways to measure change in the things that are important to families, how to best measure that. And so we're trying to ensure clinical trials are all run similarly with similar endpoints, so we can compare them to find what's truly best for our kids. That's really important information. And I'm not going to have you read this, but all I wanted you to know is that we have invested over $4.5 million into this. That's how important it is. This is where your fundraising dollars go. It goes to making sure we have all of these tools available to share with everybody, to be the low-hanging fruit. We put together a consortium of scientists and clinicians around the world to help us do a gap analysis on what is missing in Angelman. What else do we need to do? What are we forgetting? When you get to the point where you no longer know what you don't know, you're dangerous. So we never want to get there. We want to surround ourselves with expertise that we don't have to be sure that we always are doing the best for our kids and we're evaluating every single gap that's out there. We march on the Hill. We tell Congress in the US what matters. We need to tell them what's important and meaningful to our families. And we have many asks of the US Congress, and we got all of this into the language of the US Congress. We were supposed to have a patient-focused drug development meeting last month, and I was going to show you the data. because we were supposed to have that publicly disclosed on the 29th of January. But unfortunately, that didn't happen. Not my president. I'm sorry. I'm sorry to the world for that. We can control science, we cannot control that. Each platform benefits a different target. This is what's important. Each modality can lead to different biodistribution or spread throughout the brain. Different families and different patients are going to have different preferences. Single treatment therapy, needing to go under anesthesia every 2 or 3 months, wanting an oral pill and not an injection, not being able to get a virus because you have an immune response to that virus, so you may not be able to get a virus. We need to make sure we give options to families, that we find not only what's best, but what's best for you, right? That's what's important. We cannot put all of our eggs in a single basket. It would be wrong of us to do that. So we need to de-risk every option and think about the drug development process. For every 15,000 drugs developed, 5,000 get into the studies needed for humans, 10 get into humans for one to be approved. The odds are against us, people. We cannot put all our eggs in a single basket. And the cost and time to do this is insane. It can cost over $200 million to develop one drug through these phases. And this is where FAST puts our efforts. The de-risking. How can we get human data that is undeniable so that somebody responsible will take the program and get it approved and commercialized around the world? We are not here to commercialize. We are not here to do that. But we also know if we don't do it, potentially nobody will. And that's the unfortunate part of this. When Quincy was diagnosed, this is what the pipeline looked like. 2 years later, it was growing exponentially, and it kept going. It was the most exciting time in drug development. And then this happened in 2022. Biotech crashed. The bubble of gene therapy crashed. And unfortunately, 34 programs had a lot of deprioritization, and we went down to 24. But this is where we are today. We're at 27. 15 of them are fully funded by our foundation because of parents like you who support it, who see the vision, who want to be a part of de-risking things to understand how we can make a difference. 15 are funded by the foundation, which is huge. 4 clinical trials, 3 programs in IND-enabling studies, 1 about to enter a clinical trial. All 3 of those are funded by the foundation. So we are moving, and we're moving as fast as we can, but we have to be responsible, and we have to be safe, and we have to ensure we're hitting every aspect of drug development to truly be the lowest hanging fruit. So we have taken this ecosystem, internalized it, and shared it with all, to make sure everyone has the tools that they need for pharma to look at Angelman and say that's an easy choice. Because otherwise, they know that failure rate is too high. They may not choose us, but we won't let that happen. And so that is why the NIH saw this value in Angelman and gave us, in collaboration with Yale University and Rush University, $40 million to advance a gene editing strategy because they saw we had infrastructure and they believed we would actually really do it because we've proven we can and we've proven we will. And this is how Angelman gets to the top, is by putting all of our efforts to everyone and ensuring we do the right thing for all. So there are numerous therapeutic platforms that we've invested in Angelman. AS is unique. We have promising clinical trial data that we're going to hear a lot about tomorrow. We have to explore every gap in order to accelerate every shot on goal. We're not going to stop at one. It's not fair to our kids to do that. Traditional drug development, unfortunately, has failed our disease communities across the globe. That's unfortunate, but unfortunately, money talks. People have to make different priorities. But we're not gonna let it fail this one if we can ensure we support it as patient advocacy. It's very, very important, and that allows us to continue to dream big. No one's saying we're gonna cure Angelman syndrome tomorrow, that's a tagline. But what we are saying is we're not gonna stop until we make a difference in our kids' lives because they deserve that. And that's why we launched this accelerator, and I'm gonna end it here, in that Jennifer's gonna talk a bit more about the accelerator. But the accelerator was to be able to do this in a drug development way that takes this entire ecosystem and can pass it off from the foundation level to an industry level so that we can perform all of the studies that are required to get to human data, which is where you de-risk it the most. So this is what we look like now. We advanced an ASO and passed it off to an incredibly responsible party, Ultragenyx. And that inspired us to say, we could do this. We're taking our AAV gene therapy, a company that we just launched called Mavericks Bio, and we're gonna advance that into a phase 1/2 and pass that off to a very responsible party. We're gonna do it for CRISPR, a company called Courageous, because that's what we are. We're doing it for the HSC gene therapy, a company called Transformatics. And we have an enzyme replacement therapy and other programs in the pipeline. This is all your dollars at work, making sure we never stop until the data says we should. And we want to stop quickly if the data says we should not proceed. So this is how patient advocacy moves to venture philanthropy, is we have to make the impossible possible because no one else is going to do it for us. And this is unfortunately what we've learned over the years. So thank you for listening. Thank you for helping make the impossible possible. I have one reason and a horse in this race, but she's only one of 500,000. Thanks.
Thank you, Allyson, for a wonderful talk and summarizing all the projects that are going on and also reminding all of us of the perspective of the families. Thank you. We have time for a couple of brief questions before the coffee break. So I'm not going to take the privilege of the chair. Please. Over there in the back and over here.
I have a question about the accelerator. No, we know that it's very difficult and it's also difficult for a researcher's background. So my question is very open. How is going? How was your perception? And in general, how is the ongoing situation of it? Regarding the accelerator, I mean. So
what's our perspective on the accelerator? I don't understand the question.
Yes, perspective, but also your experience.
Our experience. So it's basically it's a biotech that was founded out of FAST. So the foundation fully funded it, but it's separate from the foundation. It's its own entity with its own board and its own scientific advisors and diligence team. And essentially, it is a management team of people in the industry for many, many years that are able to take human candidate to first in human and get human clinical data. And so far it's been the most amazing experience of my life, watching a team so focused on a single disorder that's multimodality. So it's modality agnostic, disease specific. So you don't get lost in what's the priority, because the priority is one, the North Star, doing the best for Angelman syndrome. And so it's really been one of the most exciting experiences that I've ever lived through, getting these programs into the clinic staggered. So the first will be the AAV gene therapy, and then the goal is to get that passed off. But the accelerator is really meant to deploy the management teams to each of these sub-companies to get each modality into the clinic.
I think there was another question over here.
Thanks for your speech. It was brilliant. So there was a slide that you were showing to us with a specific percentage of UBE3A deviance. It was 5%, 40%. So as a newly parents, because we are newly parents and we are just diagnosed our son about this, how it is possible to have this overview in terms of diagnosis of the specific percentage of this deletion or mutation?
So for mosaicism, and that's only for mosaic kids generally. So deletion and mutation and UPD, 100% of their cells are generally not expressing UBE3A from the maternal allele. This would be for mosaic kiddos where some are and some aren't. And so basically it's a blood test, which Lynn can certainly talk to you about at the break about how exactly that's done. But basically they look at the blood cells and they can see what percentage of those cells have maternal, based on methylation, have maternal expression versus paternal expression. So which has both expressing in the blood cells, which we all do, versus kids with mosaicism will have some cells with both alleles expressing and some cells only with paternal expression, showing that a percentage of cells don't have maternal expression. Does that make sense?
Yes. Yeah. Thanks.
All right, thank you. I think in the interest of time we have to move on. Sorry, but you can talk to Allyson at the coffee break. Let's go to the coffee break, and we are going to have a coffee break a little shorter, 20 minutes. Let's get back to here and start at 11, please. Thank you all.